Hi everyone, welcome back to the channel. My name is Jeffrey. Today we're going to go over the lab techniques. So lab techniques are used in a variety of ways, from research in where you're trying to test for the efficacy of a drug, for example, to physicians that are trying to confirm a diagnosis. So today we're going to go over the major techniques that you may use to help confirm a diagnosis. So let's dive right in, okay? So Southern blots, they were discovered by a guy called Southern, and it basically tests for DNA, okay? And it tests specifically for whether or not you have insertions or deletions in your DNA sequences that may lead to a certain disease, all right? So the first step that you do would be to isolate your DNA, and you can do this through centrifuging a sample, and then you're going to cut it apart using a restriction endonuclease. Okay, and what restriction endonucleases do are that they cut at palindromic sequences in your DNA, and what that essentially entails is, if you're going to look at, for example, a sequence in DNA and it's going to be CCTAGG, okay? If we take a look at the complementary sequence to that, that will be CCTAGG, right? So it reads the exact same way from 5' to 3' on both sides of the DNA. So essentially what this restriction endonuclease is going to do is it's going to cut right there and maybe leave some sticky ends, or maybe cut right here. But essentially, you can either cut like that and leave some what we would call sticky ends, quote-unquote sticky ends, or we can cut it straight down the middle. Okay, so if we cut it down the middle, it'll maybe look something like this, where we don't have any sticky ends, and this is going to be called a blunt end. Okay, and then we can put it back together through DNA ligase or something, an enzyme similar to that. Okay, so basically we're going to isolate our DNA, cut our DNA, and then after we cut our DNA, with those sticky ends that we see here, we can actually add in these things called probes. So these radioactive probes that allow us to visualize, okay? So, and that process of that probe attaching onto the DNA is a process called hybridization, okay? And essentially that's going to allow us to visualize. So if we were going to look at a southern blot, for example, maybe the normal person has, with the restriction endonuclease, they have two separate cuts, and the further down you go, the smaller the units are going to be on a blot. Okay, and then maybe the affected patient, they have a mutation here. So if they have a mutation in DNA where that this A is going to be subbed with a G, then the restriction endonuclease won't be able to recognize it, and thus it won't cut. And as a result, you may result with a larger piece of DNA that isn't cut. And that's how you would maybe say that, look, because of that, that might be proof that there's a insertion in a similar area. Okay, so for example, if we saw this type of cut right here, this type of blot right here, we would say that maybe there's an insertion at this certain gene A right here. Let's just say arbitrarily. Maybe there's an insertion here for gene A, because we don't see that on the where it's normally supposed to be right here, but we're seeing it up here. Okay. Okay, so now for northern blots are going to actually look at, if you can guess, it'll be RNA, right? The opposite, the converse of DNA would be RNA. And then if you look at northern blots, they're going to be testing for mRNA in particular, and basically it's going to look at particularly the amount of mRNA produced and the size of that mRNA. Okay, so This is a little bit easier to do than southern blotting. Basically, you're going to you're going to take your mRNA, which is a lot easier to isolate because mRNAs, you have that 5' cap, the mRNA itself, and then your poly A tail, right? So essentially what you do is you have a probe that has a poly T tail, right? And that's going to pair up with that poly A tail. So you'll be able to recognize that a lot easier. You can hybridize it a lot more easily, and as a result, you can visualize it, okay? And then moving on to Western blots, you guys are probably most familiar with Western blots. They're going to be testing for your protein. Now, the first thing you want to do is the same as the rest of them, is to isolate your protein first. You can do that through a centrifuge, for example. And then you want to break apart those disulfide bridges. Remember, in tertiary structure, you have a bunch of disulfide bridges, right? So you want to break your disulfide bonds, and you can do that through this chemical, and it's going to be called mercaptoethanol, and that's going to be one of those chemicals that are very commonly used. If you've ever worked in a lab, you would know that you use that to break those double bonds. And then you would basically put some soap over it, and that's going to be called your SDS, your sodium dodecosulfate. Basically, this is the same thing as just normal soap. But it's going to give that protein a negative charge. Now, the reason why you would want to give a protein that negative charge is because, remember, your protein has positively charged and negatively charged areas. For example, like lysine, or like your glutamic acid, right? So in comparison to your southern blot and your northern blots, which test for DNA and RNA, remember that DNA and RNA both have this negative charge because of the phosphate groups, and they don't have any other charges, so you don't need to add something like sodium dodecosulfate, okay? So you add in the SDS, and then what you want to do is you want to add in a primary antibody that's going to bind to your protein of interest, right? Binds to the specific protein that you want to take a look at. And for the most part, a lot of these primary antibodies, they do not glow, or they do not fluoresce under a microscope, or any other visualization technique. Sorry, I just butchered the spelling there, but anyways, you're going to have typically a secondary antibody that you add on that's going to bind to that primary antibody, but this one is going to be attached to a fluorescent probe, okay? So you can actually visualize your protein. Now, if we were to look on this on a blot, it might look something like, if we were going to look at, for example, familial hypercholesterolemia, a normal patient might have, and if this is what we're looking for LDL receptors, we might have a lot of LDL receptors. And then for a person with familial hypercholesterolemia, we might have a much smaller band. So the magnitude of that matters. So it's basically, Western blots are measuring the amount of protein that you're producing, okay? And then we have this thing called sequencing. It was discovered by this man called Sanger. That's why we call it the Sanger method. But basically, this is going to be sequencing your DNA. You're going to take a look at directly what that DNA sequence might be. And so essentially, the first step that you do is you denature your DNA. And after you denature your DNA, to expose that template area, so if we have that DNA template up on top here, on the bottom, what we're going to do is we're going to add in some radioactive dideoxynucleotides, okay? So we add in the radioactive dideoxynucleotides, and that's going to, and then we form 4 aliquots. Okay, so each aliquot is for each nucleotide. So we have 4 aliquots, each is going to be, you know, A, C, T, G, stuff like that. Okay, and then these dideoxynucleotides, remember that your normal nucleotide looks something like, something like this. You have your R group here, you have a phosphate group up here, and then you have your OH. This is a deoxynucleotide, right? That's a DNTP, but DNTP basically just subs that out with an H, and because you don't have that free OH anymore, you're going to have early termination. You won't be able to synthesize anything more. So that allows you to be able to actually, sequence your DNA because you won't be able to polymerize anything. Okay, so essentially you're going to have your 4 aliquots. Remember, A, T, C, and G here, and then you're going to read from the bottom up because remember the smallest fragments are on the bottom. So if we had a blot that looked like this, we can safely say that the DNA sequence would be A, C, T, G, T, A, C, T, G, T, right? And that is going to correlate with my DNA sequence. So remember, this on top was my template. So on the bottom is going to be where my my actual sequence is going to be. Okay, so that's how you sequence using the Sanger method. And then there's a test called RFLP, which is called restriction fragment length polymorphism. Basically, there's 4 different things that that you can do with this. Okay, so this is looking at basically the polymorphism of one base pair. So if that one base pair changes, where it's going to change your restriction site for your restriction endonuclease, and thus it's not going to cut there. Okay, so this is usually only used for known mutations because if we don't know what the mutation is, then it would be impossible to test for. Okay, so only for known mutations. Okay. So the first way you can do it is through southern blotting. Basically, you isolate your DNA, and then what you're going to do is you cut it up with a restriction endonuclease like you did before, and then you add in a radioactive probe to that area that you cut. Okay, and then you just compare the sizes. All right, so yeah, so if you have different probes, then you can see different lengths on your gel. And then for PCR with restriction endonuclease, basically this is a newer technique, and you're just taking your PCR of a very small fragment that you're going to look at. You digest it, so you PCR a short fragment, okay? And then you're going to use a restriction endonuclease to digest, and then you're going to visualize using ethidium bromide. That basically just stains any DNA and allows you to visualize it on an agarose gel. Okay, and so moving on, ASO is going to be your allele specific oligonucleotide. So that sounds really complex for something that makes a little bit more sense, because for this, it's going to be a common mutation. So if you know that, for example, maybe a CTG is going to result in from a CAG is going to result in maybe a disease like, if we're just going to talk about cystic fibrosis, for example. If that results in cystic fibrosis, then you can use your allele-specific oligonucleotide to test for these sequences. And essentially, you don't have to do anything, okay? So essentially, all you got to do is just take the DNA, denature it, add in the probe, and if you see a speck, if you see anything, you have the disease for that probe, okay? And then for ASO PCR, essentially you're doing the same thing. But remember, this is probably going to be a speck on your agarose gel. Essentially, you're just going to, with PCR, you're just going to magnify the speck. Okay, so this is just going to give you a much larger or a much more easily seen probe. Okay, so I hope this helps. I hope these lab techniques, my description of these has helped you in some